And but you don't really, you only see people for a split second because then I switch to the screen. That way anybody who is absent can watch the class and not just get the lesson, get any conversation that occurred. And that's how they get we do credit for class participation, and that's how they get credit for class participation is they comment on the video on our YouTube channel. We've got a public YouTube channel, and then we have a private YouTube channel that is not advertised. They have to have you have to have a direct link to get to it. So that's how they so that's how they watch if they're not here. Do I have any quarters? I might later though. Do you want me to look? I'm gonna hold on to them for you and look later. Cool, awesome. I'm gonna put them right here. So we have pennies. This is actually a little box. These are little ice cube trays. They have everybody's name on them. And how do you get pennies, girls? You answer questions. If you answer a question, even if you're wrong, you still get a penny. If you contribute to the conversation, you get a penny. And then what do we do with the pennies? What do you do with them, Everly? What do you do with the pennies? Everly's very quiet until she doesn't want to be. Then she can be quite chatty. They go in the Skittles machine on the way out the door. Well, this is how we encourage people to keep talking, stay involved, stay active. And we're just waiting on Evan. So these two always are in here a little early because that's Miss Jackie up there. So Jackie does Spanish. So they do Spanish with Jackie. And then they hop over here. And now she has another level Spanish class. That's her Spanish 2. Y'all are in 1, right? That's Spanish 2 or 3. I don't remember which one that is. I think it's 2. And she teaches them in there. And we always have done this for 8 years. We've been trading students. So it's great because it's a one-stop shop for parents. You can get 2 of your classes done at one time. It's very helpful. And Colin over here, he does Spanish with them, but this is our juniors class and he's in our lab rats level class. So he just sits this class out and like does his Spanish homework or whatever he needs to do until it's time for them to move in. This class is shorter. It's 45 minutes. His class is an hour and a half.
So like elementary to middle school level?
Yeah.
Okay, just was wondering what junior meant.
Yeah, that's exactly right. These guys are like upper elementary to middle school. You girls are what, 10, 11, 10?
Oh man.
They're twins. So yeah.
I was gonna say I could.
Yeah. Yeah, they're twins. So, and I had their older brother a couple years ago. I had them last year too.
Do you mind if I interview you after 1:30?
Absolutely.
That's
fine.
like,
You're good.
So many questions.
Yeah, I've graded all the things I could grade ahead of time so that we would have some time to chat. So after class every week, I send out an email just once a week. I try not to bombard them with anything during the week if I can avoid it. Things pop up occasionally. Like I send out an announcement that you would be here. Like we're gonna have a guest because I want people to know when they walk in and they're like, who's this weird person? You know?
I hope I'm not.
No, we like company. We just don't get it very often. So I'd be sure to tell everybody, expect company. Absolutely. So they don't walk in and be like, like when my dad's here, they all know to expect my dad's visiting. So yeah. But yeah, we just meet the 1 day a week. So we cover they read before class, they have a lesson. I can show you when we chat, but we have a syllabus that they get the before class starts and it has every assignment from now to the end of the year, like a college syllabus would be. There's no guessing what's coming next. They know exactly what it is and when it's due so that if they want to work ahead, they can. They also have a final project every year. We have a different final project and that's assigned first day of class. So they have all year and there's a rubric. They know exactly what they need to do. So we don't have any, but inevitably though, we have, 2 days before it's due, panic attacks because we had all year, but we're going to wait to the last minute.
Yeah.
Because why
not? That's going to happen.
It happens every time. Yep. But yeah, these two will be starting middle school next year. So you'll be going to public next year, right? That's been the pattern for their family. So they have 2 older siblings that did the same. Did Cameron? Elementary? Yeah, okay. So elementary is homeschool for their family, and then they start public school in middle school. I think they're the only ones right now that in this group that does that. Everybody else is long haulers. So people do it differently. I've known a couple different people who've done it that way. Come on in, Evan. You'll get a chance to look at them after class. You're good. They're over there on that in that big stereoscope, and we'll make sure you get a chance to look at them before they get hitched. Erin, this is Miss Macarena, and she's just here. I mean, sorry, Evan. I said your mom's Erin. I was thinking about your mom, Evan. This is Evan. And so this makes our class. This is a small class. The next class is a big class. The juniors class is always smaller because they don't need credits. The high school kids are trying to get high school credits.
Oh,
So
yeah.
I always have a lot, and chemistry is always the biggest class because that's the one that parents are least likely to want to teach themselves. So there's always a waiting list with chemistry. It's always absolutely full. That was last year. We had an absolute full load and 8 on waiting list. So that's the challenging one. This one, this biology this year. So we got biology, and then next year's earth science, and then physical, and then I'll be back to chem again. So I rotate the 4, but next year I might start doing 2 days a week if there's enough demand, and I would offset by 2 years. So it's bio, earth, physical, chem. So next year it would be earth and then chem, and then the following year would be physical and bio. That's my idea, at least. We'll see how it goes. We are the only program in this state that's anything like this. Nobody else does what we do. There are
I
some,
thought that was really cool the
Yeah.
line you sent. I did.
yeah, there are some co-ops that do, biology and whatever, but I'm not aware of anyone else that does weekly labs, and we do. And a lot of homeschoolers do what they call like kitchen chemistry. It's, all the basic little things that you have at home, or maybe order this little thing from Amazon, but we have all the gear. Like we have, like you can see, real microscopes. That one, all of ours hook up to digital cameras. We've got all of the high-end glassware, the burettes, the stuff like that make it impossible for homeschool families to do on their own unless they have a lot of expendable income, because it's very pricey. Like the burettes are $30 a piece, and that's just one tiny glass thing. So when we all get together, we share the cost. All right, are we ready? Binders open. So they've all done their reading, and they've gone through their binder and filled out their binder pages coming into class, so they know what they're doing before they get here. So hopefully that will help them answer some questions. So we are now in second quarter. Today starts second quarter, right? That means you'll be getting a progress report from me once I have all your assignments. So if you look, and if there's anything left that you haven't done, you've got just a few days to squeeze it in. There'll be a late penalty, but you can still squeeze it in. It's better than a 0, right? So, all righty. And then we're starting a new unit. This is unit 4, it's genetics. And this is a really fun unit. This is one of my favorites. All right, so let's have a look. So the first thing we're going to talk about is this dude, Gregor Mendel, and he is seriously one of my favorite scientists. He's a really neat guy. So we like to talk about our scientists as people, so we understand the context in which they were living and studying and learning. So this is Gregor Mendel, this dude right here. So we're talking mid to late 1800s is when he lived, and he was a polymath. Does anybody remember what that word means? We've had a couple of polymaths in the past. Anybody remember? A polymath is somebody who's very good, very well educated in multiple different disparate topics, right? Things that may not even have anything to do with each other, like Benjamin Franklin and Leonardo da Vinci and Thomas Jefferson, and Alexander Hamilton called himself a polymath in the musical in one of the lyrics, right? Yeah, you girls know that because you're musical people. So what do you suppose were his areas of interest? We'll take a guess. Take a guess. What do you think, Evan? What's something he might have studied? Besides biology? Could be anything.
Cells.
Cells, yeah, okay. What do you think, Everly? Take a guess. What's something he might have studied? Anything. He could have studied anything. Nothing? Could be knitting. It could be ice cream flavors. What do you think, Everson? Anything? Chemistry? All right, so let's see. So his areas of interest were meteorologists. What do meteorologists study? Weather. Mathematician, we know what that is. Biologist. He was an Augustinian friar and an abbot of St. Thomas Abbey in this place, Brno Monrovia, which is now in Slovakia. And I'll tell you what, my husband and I took a trip once, and we drove past that town on the highway, and we spent like 30 minutes trying to figure out how to pronounce it because there's no vowels until you get to the end. Like, is it Brno? Bruno? It's almost like Bruno is how you say it. Because I have a friend who actually lived over there, and she helped me with that one because I was like, I have no idea how you say this word. So he was born Johann Mendel, the son of a struggling what? What do you want to guess his dad did? Farm. Very good. He was a farmer. Good guess. You're right. He was a farmer in a German-speaking family in what today is, what country do you think? close to Germany, kind of. So today it's Czech Republic. The Czech Republic has been through several different names in the past. Czechoslovakia, probably most recently, but the Czech Republic, which is a beautiful place. If you ever get to go, do the Czech Republic. So then he became a monk, in part because it enabled him to obtain something without having to pay for it. What do you think monks get that they don't have to pay for? There's lots of things. What do you think might be important? You've studied monks? Yeah. So he was able to get an education without having to pay for it. That's a big deal. Okay, because education beyond everybody back then was homeschooled, right? All these great scientists in history were all homeschoolers. But then when you get to like adult age and you want to continue your education, college was expensive. But if you become a monk or go into somewhere into the ministry where you're living a monastic life or you're clergy or you're a priest, you get educated. So the monastic life, in his words, spared him, this is a great quote, the perpetual anxiety about a means of livelihood. Now, keep this in your brains. This is something to remember kind of forever. But a lot of our big historical scientists, our big scientific celebrities of the past, were men of the cloth. They were monks or priests. And the other option is independently wealthy. Now, why do you think that is? Why do you think a lot of them weren't farmers or shopkeepers or factory workers? Why do you suppose it's monks, priests, and independently wealthy people? What do they do all day?
Study.
They have time to study, absolutely. And what do they not have to do all day?
Work part.
Amen. They don't have to work all day. So because they don't have to work all day, they have a lot of time to think about stuff. Also, they're all educated. Independently wealthy, you can afford the education. Monks and priests get educated by the church. So they've been taught how to think. And they're people who are geared towards really deep thought. Why is this canonized and why is this not canonized? Why was this verse in the Bible and not that verse in the Bible? So they've done a lot of thinking. So these are people that are geared towards thinking, and these are people that have lots of free time to think. So a lot of our greatest scientists in history have been monks, priests, or independently wealthy people. So he gained posthumous recognition. Do you know what posthumous means? That was a little harder to break down into its parts. That means after you die. Okay, so not during his life, but after he was dead is when he gains recognition as the founder of the modern science of what do you think? It's what our unit is on. Genetics. He's our founder of modern genetics. So he experimented with pea plants, and I'll give you the years. It was like the middle of his life, his middle age, so the 1850s and 60s, and established many of the rules, and we now call this laws of Mendelian inheritance. Mendelian coming from his name, Mendel. So remember, he was born named Johann Mendel, but then when he became a monk, they gave him a new name. That's Gregor. So now he goes by Gregor Mendel, but we use his last name, Mendel, in the word Mendelian inheritance. So cool, huh? So he coined these two terms that we'll use a lot, and those are dominant and recessive in reference to certain traits. He published his work in 1866 and demonstrated the actions of these invisible factors that we now call what? What do we call the things that determine our traits? Whether you have dark hair or light hair or blue eyes or DNA, but what do we call, it's in your DNA, but what do we call those little chunks of DNA that determine? We call those genes. So he didn't have quite the word for that so much, but he knew there was some kind of invisible factor, something he couldn't see that was controlling how traits were inherited. So profound significance of his work was not recognized until the 20th century. So more than 3 decades after he was dead, people are like, hey, this guy was onto something. Such a shame that during his lifetime, people weren't, yeah, nope, after he was dead, and ushered in the laws of modern genetics. All right, so let's have a look. This is what he studied. Pea blossoms, pea plants, like the kind you eat, okay? And he studied the blossoms. First thing to understand is to get kind of your basic anatomy of a flower. And we'll do this more when we do our unit on plants. But there are different parts of a flower. See, you can break it all down. And what it's showing here, all these are all the reproductive structures of a flower. What allows a flower to get this little ovule fertilized, which will then usually grow into a fruit of some kind, and then it can reproduce, right? Because that's where the seeds are. So parts of the flower are the male parts, and other parts of the flower are the female parts. So I'm going to show you which is which, so that you kind of know. what's going on. So over here, the anther and the filament, this is where you find the pollen, right? You ever looked at a flower and you've seen the pollen on those little anthers, little yellow dust or green dust? That's the male part. And then over here, all these parts are the female parts. Down here is what they call the ovary, and then that's the ovule. The ovary is what's going to develop into the fruit you eat. Like, say this is an apple or a pea. And then the ovule would be like the seeds in the apple, for example. Okay? So that's your basic flower. So what do you call it when a new cell forms, when a male and a female gamete, so like an egg and a sperm or sex cells join? What's that process called? When an egg and a sperm come together, you get, it starts with an F. Fertilization. So you get fertilization, go on. When you get a male and a female sex cell come together, you get a new cell with new genetic makeup. That's going to be fertilization. So in plants, there's two different ways. You can self-pollinate and you can cross-pollinate. So when you self-pollinate, it's when you've got your own pollen going into the tippy top of that pistil, and then it'll go down that pistil into the ovary and fertilize the little ovules. All right, so there you go. That's self-pollinating. That means it's the same genes reintroducing themselves to one another, right? So this is not going to be genetically different. The plant that it produces will be genetically the same as the parent. But when you cross-pollinate, which is what bees do and butterflies do, and sometimes hummingbirds even, right? They're going to take the pollen from one plant and put it on the pistil of another plant. And now you're getting some genetic variety. Okay? So plants can go asexual reproduction and sexual reproduction. They can go both ways. So pea plants are usually self-pollinating. So pollen from a flower lands on the pistil of the same flower, just like that, like we showed you before. And then some of them, though, Mendel created a way that he could cross-pollinate pea plants. So he took a little tiny paintbrush and he would collect, like a bee might, pollen from one flower, from the anther of one flower, and then put it in the pistil of another flower. And just to be sure, he would remove the anthers from that flower before he put the pollen on it, just to make sure that none of its own pollen got into it, right? So he's cross-pollinating. So a purebred organism is the offspring of many generations that have the same traits. And then Mendel started his experiments with purebred plants. So we know the genetic makeup of these plants. We know what to expect from their offspring, basically. So he studied 7 traits in pea plants. So he looked at their seeds and he said, are their seeds round or wrinkled? Is the cotyledon inside the seed, that's like if you break it open, you'll have split pea soup, split the pea in half. Is it yellow or green? Then he studied, are the flowers white or violet? Are the pods full or wrinkled up and constricted? Are the pods green or yellow? And then where are the flowers on the stem? So it says axial pods and terminal pods. Axial means they come along the branch. Terminal means they grow at the end of the branch, see? And then size, was it tall or short? So he studied all of these 7 traits, all right? Okay, now, here's a quick TEDx, we'll throw this one in, that kind of sums up what I was just telling you about what he did. With good animation. We like good animation.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase y always overpowers its lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes.
Did it go out?
Two possible phenotypes in a 3-to-1 proportion.
You know how that is. Sometimes it'll do that. I have to kick it back on. There you go. And doo-doo. Uh-oh, hold on. Gonna do its thing now. There it is.
But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations. Round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
All right, so now let's talk a little bit about these laws of dominance and how they work. So genes pass, what do we call the characteristics that get passed? Starts with a T. You have it in your binder. Does anybody have it? No? Okay, traits from parents to offspring. Different trait forms. So it said it in the video, different forms of traits. This is this fun word, allele. Okay, these are alleles. All right, now, in the most basic form of Mendelian genetics, which is what they were just showing you, each trait is derived from how many alleles are involved? How many did it show? How many Y's? How many R's? 2. Okay, a pair. That's what we want. A pair of alleles. Each allele is categorized as either dominant or recessive. And here's the trick to remember. Dominant covers recessive. Covers is the word. And the reason covers is the word is because that recessive allele is still there, but it can only be expressed or show itself if there's not a dominant with it. Because if it's paired with a dominant allele, that dominant allele is going to cover the recessive one, okay? So dominant alleles, what do? They cover, just said it, they cover the expression of the recessive alleles. The dominant allele is then expressed. So then, a combination of alleles, two alleles together, that's going to be a genotype. Okay, what type? Genes. All right, genotype is the combination of alleles. All right, so it's what your genes say, but how the trait is expressed is called what? Not genotype, but it's very similar and it rhymes. Did anybody catch it? This is phenotype. So this is, you can think of it as how it looks or how it's expressed. Did you forget your glasses today? You keep squinting. Oh, goodness. So yes, so your genotype is what your genes say, and your phenotype is how it's expressed. All right. So for example, if you have a say you have a parent with blue eyes and a parent with brown eyes. Brown eyes are usually dominant and usually cover blue eyes. So you could have a brown eye allele and a blue eye allele, and your eyes are going to show as brown. So your genotype would be a dominant and a recessive, but your phenotype is going to show what the dominant is. So it's going to show brown. And we'll look more at that. So dominant alleles are designated with what kind of a letter? Lowercase or capital? Capital, right, Emerson. Good. Whisper a little louder. There you go. And then your recessive alleles are going to be shown with what? Lowercase. All right, very good. Simple. So if you look here, dominant is they're giving it an A, and I assume that A stands for, what do you think that A means here? What's the difference in these cherries? What's the difference between this pair of cherries and this pair of cherries? They're different colors, right? So what do you think that capital A stands for? What color? Red. It's red, right? So in this case, you have a dominant and a dominant, two dominant alleles, and you're going to get red. You have two recessives, and you get what color? Yellow. And then here, look, you have a dominant and a recessive, but what color do you get?
Red.
And that's because the dominant does what to the recessive? Covers it. You got it. You got it. All right. So the principle of dominance states that dominance always exhibits the form of that trait. So your capital A, your lowercase a, if you've got a capital there, the lowercase is not going to show, right? That's going to be your phenotype. Recessives always exhibit the form of that trait only when there's no dominant present. No dominant present there, so you get the yellow. All right. Monohybrid cross. So here's amoeba sisters, and that's what we're talking about. We get an ad. We're talking about monohybrid crosses right here. We're just talking about like one little pair of alleles.
So I have a confession to make. For my classroom, I always wanted a hairless guinea pig. They sell them at some pet stores, but
You're
they
creepy.
can be really hard to find. And while I doubt they do so well in the wild, their genetics are fascinating. And so are guinea pigs. We spent quite some time on LOL cats, but we'd spend even more time if someone would make a funny guinea pig site. Most guinea pigs have hair. This is because of their DNA. DNA makes up genes, and the guinea pigs received genes from both their mother and their father. An allele is a form of a gene and often represented by a letter. In this case, we'll use the letter H for hair. A hairless guinea pig has two recessive alleles for the trait of having or not having hair. A recessive allele is usually represented by a lowercase letter or allele. By recessive, it means that the allele will not usually show up. The only way it will show up is if there is no dominant allele present. A dominant allele is represented by a capital letter, and it's an allele that will show up. Think of it as dominating. So a hairless guinea pig does not have a dominant allele for hair, and that's why the recessive trait, not having hair, shows up. A hairless guinea pig's genotype, that's the genetic makeup of an organism, can be represented as a lowercase little h. A guinea pig that does have hair can be represented as a big H, or a big H, little h. See, it only takes one dominant allele, a capital H, for a trait to show up. That dominating allele means that the recessive allele is hidden. Now, we're not quite done with the vocab here. A genotype of big H, or little h is considered homozygous. The root in this word means same, and they are the same case. Big H, those are both capital letters, and little h, those are both lowercase letters. So big H is called homozygous dominant because of the capitals, and little h is homozygous recessive because of the lowercase. A genotype of big H, little h is considered heterozygous. The root in this word means different because their letters are different cases. One is a capital and one is a lowercase. If I were to show you a guinea pig with hair, would you know its genotype?
Would you?
Well, you would know it's not little h, because
No, which
if it was
one's
little
not?
h, then it would be hairless. But remember, a guinea pig with hair, it could be big H, or big H, little h. We don't really know. We could do a test cross to eventually determine this, but you can't tell just by looking at the guinea pig. Now, if we had a hairless guinea pig, we do know
Would
for
you know
sure that
here?
it's little h,
Yes.
because if it did have a capital letter, even one, that dominating allele would take over and it would have hair. So let's try a monohybrid cross. The root mono means
I'm going to pause this one here.
one because
You can watch the rest of it. You know, I put it in the resources. You can watch the rest of it later, but I wanted you to watch that part one more time so it's been explained twice. All right, so now let's apply some principles. By the way, this is Mendel's research garden when he was in it, And it's been preserved today, but those look like begonias, not peas. But it's still there. You can go visit if you want to. So you've got a trait for height, let's say. Now, in humans, it's not one or the other. Not everybody is 6 foot or 5 foot, right? We have a range, and that's different. And we'll talk about that later. But in pea plants, they're either tall or short. There's kind of nothing in between. All right. So you've got T and T we're going to use. Capital T for tall, dominant. Short T is recessive, and lowercase t. So here are genotypes. Capital T, little t, and then capital T, little t. What do you call this type of genotype if it has 2 dominant alleles? It's something zygous. What was the word that means, the root that means the same? What's same? Homozygous, and then is it dominant or recessive if it has 2 capital letters? No. Yeah, you say it. Dominant. There you go. So that is homozygous dominant. What's the next one going to be? What do we think? Homo or hetero? Homo, because it's little t. They're both the same. And is it dominant or recessive, Everly? They're 2 little letters. Very good. Okay. And then what's the bottom one going to be? Homo or hetero? Hetero. And then what is it going to be? Dominant, or it's just going to be heterozygous, right? But it's going to show which trait, the dominant trait or the recessive trait. The dominant trait. Yes, you guys got it. All right. So this is what you get: homozygous dominant, homozygous recessive, and heterozygous, which is obviously going to show the dominant trait. Now we have to talk about phenotype. This is genotype. What are the genes? Phenotype is what does it show? What is expressed? So if you have 2 capital T's, a homozygous dominant, and capital T means tall, what's the plant going to be? Tall or short? What's it going to be? Capital T means tall, and you have 2 dominant tall genes. So is it going to be tall or short? Tall. Good job. All right. How about the next one? 2 little t's. Tall or short? Short. Very good. And then how about the last one? What's it going to be? Tall. Why is it tall and not short? Because it has a dominant allele, and it's going to do what to the recessive allele? Hover it. Very good. You got it. All right, now let's talk about chromosomes because these alleles are parts of genes and they live on chromosomes. So chromosomes are in pairs. So if you look at these in your head, you can probably kind of start matching these pairs, right? You can tell what goes with what, right? Like, let's see, there's that little funky sandal with the toe thing and that one there too. And then these shoes go together, and then lastly these. Now they're pairs. And here's why to remember that it's important to remember that they're pairs. We call these homologous pairs. They're the same, okay, but they're not identical. And here's what we mean by that. They exist in pairs. Look at your shoes, like for real. Look at your shoes. Are they identical? Are they? Do you have a left and a right shoe? So are they identical? Can you put your right shoe on your left foot? No. So they're not identical, but they're a pair, right? So that's how they compare to homologous chromosomes. They're identical, but not, they're pairs, but not identical. We call them sisters, right? They're similar, but not identical. You have a left and a right, so they're not identical. And in all the shoes in the class, look at everybody else's shoes. If we threw all the shoes in a pile, do you think you could pair them up? Yeah. Even if you threw in not just you 3, but Macrina's and mine, and we threw them in, then we'd what? We'd be able to tell which goes with which, right? But they're still not identical. They're pairs. So no 2 pairs are identical either, right? Well, the pairs are different, but all the shoes in the class are like unique pairs of homologous chromosomes that are in your cells. All right, so they exist in pairs, but they don't really look like shoes. They look a little bit more like this. Have you ever seen chromosomes shown like little X's? Yeah, you probably have. All right, so they match, but they aren't identical like pairs. And this is because one chromatid, and you can think of a chromatid as one shoe, comes from a male parent, and the other chromatid, the other shoe, comes from the female parent. So if you look in this case, you've got kind of like a bluish one, like a pinkish purple and a bluish purple. So let's just call them blue and pink for the sake of not being confused about purple. So the blue one comes from the father and the pink one comes from the mother, and you can see that they line up, right? They look almost like shoes that would go together, but they're not going to be identical because the alleles on those chromosomes are going to be different. So each chromatid has a series of alleles, and its sister chromatid or its pair, right, is going to have the same series, but the alleles might be different. So if you look there, you see that you've got an allele for blue eyes and an allele for brown eyes, but you see how they're in the same position on the chromatid? You don't have the brown eyes not down here and the blue eyes up there. They're side by side. They're looking right at each other. So that's how it works. Does that make sense? All right. So it's pairs within pairs. So during meiosis, now last week we talked about what's the name for cells dividing and making more cells? No, started with an M. Mitosis. Right, that's mitosis. Okay. Unfortunately, this sounds the same, but it's meiosis. Okay, very similar. Meiosis and mitosis are different. Mitosis, the cell divides and makes two identical cells, right? Meiosis is going to start the same way, but you're going to end up in the end not with two cells, but with four cells. And each of those four cells is going to have half of the genetic material of the cell it started from. And this is how gametes or sex cells are made. Sex cells, gametes, eggs, sperm, only have half of the DNA. We call them haploid. If you have all your DNA, you're called diploid, okay? So it's gamete production, and in this case, you get what we call chromosomal segregation. And I'm not going to get too picky on this with you guys at this level, so we're going to kind of breeze through these, all right? So just kind of follow along, but don't worry too much about like getting every little intricacy. So you get what's called chromosome segregation. So if you look here, you've got 3 pairs of chromosomes. 1, 2, 3. Okay. Now, in this case, you've got your chromatids, right? Chromatids come together. You got a chromosome, you got another chromosome. And if you look, you see how they can divide up. This would be one cell containing all these three types of chromosomes. And in the end, look what you end up with. You see how they're all different combinations? Okay. So that's how we get such genetic variety. That's the big thing for you guys to remember. We get all this genetic variety with sexual reproduction, and that's why, because chromosomes are independently assorted. So the principle of dominance predicts each phenotype is determined by 2 alleles, which we said, and the dominant allele is going to do what? It's going to cover the recessive, right? So that's one of Mendel's laws. Another one is the principle of segregation, and this says that during gamete production, each gamete receives one of the 2 alleles. So one of the 2 alleles from your parent. So a parent might have an allele for brown eyes and blue eyes. Okay, so they've got a capital B, little b situation. But during meiosis, they're going to create gametes or sex cells or sperm or egg. One of them is going to have the big B and one of them is going to have the little b. So their own genetic material is getting divided up, right? And then they're going to combine that genetic material with that of another individual if it's sexual reproduction. So chromosome segregation predicts that basically this is saying it's kind of random, that each gamete receives one of 2 alleles present in the parental cell. And then this is the random one, sorry, independent assortment. So predicts the alleles coding for different phenotypes are segregated independently from one another. So it's just showing you can get all of these different combinations just starting with the same cell, right? Can be sorted many ways. So this should be in your binder. So let's see, let's have a look and let's see if we got it right. So for number 1, it's showing probability of producing offspring that have short whiskers from a cross of 2 long whiskered seals. One is homozygous dominant and the other one is heterozygous. So let's see. You're going to start with that. Your heterozygous dominant, is it going vertically or is it going horizontally there? You tell me. Which one is your heterozygous dominant? Heterozygous. So that means it's different. So that's, see how that was the horizontal one? Capital W, little w, right? And then your homozygous is going vertically there. You can put them either way. It doesn't really matter. I do suggest that if you're doing like male and female parent, that you pick a pattern and stick with it. Do female across the top and male down, or however you want to do it, but just stick with it for your own sake. So you end up with this combination. So you combine capital W, capital W, capital W, capital W, capital W, little w. See how that works? And what percentage of your offspring are gonna have long whiskers? Somebody tell me what you got. What'd you get, Everly? 50. And what, oh, no, 100%, sorry. So what'd you get 50? What made you think 50? Long whiskers.
Yeah.
I think that's supposed to be right. Wait a minute. No, long whiskers, 100%. So here's why it's 100%. So if you have that capital W, it's gonna cover the lowercase w. So you have 50% have the gene, you're right, that 50% have the genotype for dominance in both cases. But when you look at how it's expressed, remember that big one's gonna cover a little bit. That was a good guess though, really good guess. And you're gonna have no short whiskers. So let's look at the next one. You have a heterozygous long whisker and the other is short whiskered. So what's the genotype for the short whiskered one gonna be? Gonna be 2 little W's, right? Because it's recessive. So heterozygous, he's gonna have long whiskers across the top, but his genotype is long short, right? But he's gonna show long because long's gonna cover short. And here he has short short. So let's see, how does that turn out? All right, what now tell me what percentage, Everly, this time? What percentage are gonna have long whiskers this time? Did you get this one? Not sure? What'd you get, Emerson? 50 and 50. This one's 50. You maybe read that one. Looked up before. 50 and 50. There you go. You see why? Does it make sense? Yes? Okay. And then this last one, let's just check your answer. You should have that. That should be kind of what your answer is. Horned, unhorned, right? All right, making sure that you're checking it. All right, so then when it comes to a pedigree, so you had this one too, huh? Do you have this one? Find it? There you go. All right, so what's the name of Irene and Leo's son? And what's the name of their son-in-law? Anybody have an answer? Do you have this? What is the answer for number 1? Anybody have it? Not sure? Okay, so Irene and Leo, who is their son? Gordon is their son. And then Tim is their son-in-law. Oh, wait, that shouldn't say Richard there. That's wrong. I have to fix that. Emily's their daughter, Gordon. Why does that put that Richard there? Hold on. I'm going to correct that. That's not supposed to be there. Tim is their son-in-law. Now, there it's fixed. All right. And then how many grandchildren do Irene and Leo have? Do you have an answer? Anybody have an answer? Emerson? What'd you get? 6? Yep, 6. And then how many of them are girls? 3. Okay, what's the name of Ralph's father? Who's Ralph's father? Anybody have that? Gordon. Very good. And Ashley's mother is Cheryl. See? It's good that you learn how to read these because we're about to start that cold case lab today, and this is how it ends up. You're going to have to follow a family tree along, right? So what's the name of Emily's son? Here's Emily. What's the name of her son? Richard Jr. Very good. And then Tim's son is going to be Zach. He's over here. So after this pedigree was made, Richard and Emily had another son whom they named Roger. Juliet married a man named Robert and had a daughter named Elizabeth, and Zach married a woman named Jean and had a son named Craig. So if you were going to finish that out, you've got Roger, Robert, Jean, and Craig. Got it? So it should look something like that. All right, and here are these. I want you to just look. I'll give you a minute to just check your binder, check your answers, because we have an activity to do. So I want to have time for that. And your answers are there. Check them over. You can always come back and look at it, right? We know we can come back and look at it. All right, so now we're going to breed dragons. All right, here's what we're going to do. I'm going to give you each a worksheet, and you're going to start today by you're going to get a dragon. You're going to build a dragon of your own. Okay, you're going to get the alleles, and you're going to build out the dragon. And then over the next couple weeks, you'll keep working on it, and you're actually going to breed your dragon with somebody else's dragon, somebody else in the class. And that way, then you're going to have a baby dragon with different genetic makeup. So where we're going to start is I'm going to give, okay, hang on to this little bag. You're going to put your alleles in here. And here is your worksheet. And you can leave your worksheet here for a minute, but you're going to take your bag with you. Everly, here's yours. Whoop, and Evan, here's yours. Take your bags. You're going to come up to the counter and you're going to randomly, don't look, randomly take 2 pieces of paper from each cup. Okay? Go grab them. Go for it. Take 2 pieces of paper from each cup. It's going to be your alleles for your different characteristics. And stick them right in your bags. Do they look good, Kat? They're cool, right? They're all alive. Yeah, and you've got tails that are growing eye spots and heads that are swimming on their own. Love you. Have a safe trip.
Friday, let's say Thursday.
Oh, Thursday. Okay. All right.
Yeah.
Love you. Bye. Claire, I like your haircut. So after you've got them, keep moving down so everybody have a chance to grab 2 strips from each cup. Yeah.
All right. Now, right here.
And while you're doing that, Emerson, I'm going to look for a quarter for you because I think I have a thumb here. Trade back the pen for you. Here, I knew I had one. Randomly on the counter. I'll put the expired water bottle, okay? Thanks for the pens. Trade bigger coins for littler coins so that we always have Skittles money because the machine will take any coin, but pennies go further than quarters. So there we go. Awesome. So when you've got them, come sit back down. So this is what they're like. I got plenty of them. Yeah, sure. Got that one, Emerson. There you go. Good job. Excellent. Once you've collected them, I'm going to take the numbers. All right, thank you, sir. Very good. Have a safe flight. Enjoy after this evening. Guys, go to Tulsa with the big Claire wants to go.
Yeah. it's fine. Good time.
Big us to over this. Next one. Going. There. Okay. There. Excellent. Okay, take those back to your seat. Don't lose your bag. Hang on to your bag. Awesome. I'm just gonna stick it. Oh, so easy. All right. So here's what I want you to do. Look at your handout. So work on this kind of on your own when you have time. We'll have time in class, but so don't lose it. So like maybe put it in the little pocket in your binder so you don't lose them. But what you're gonna do is each of those strips of paper was an allele. It'll have a capital letter or a lowercase letter. All right, and you're going to take your pair of alleles, and you see this right here, this table. So you're going to enter whether you have a big B or a little b, a big D or a little d, and it'll tell you on that allele what the characteristic is. Like it'll say, big B is a nose spike, little b is no nose spike. Big D is wings, and little d is no wings. Okay, so you're going to write out what your genotype is. So you might have big B, little b, and then your genotype is big B, little b. So it'll be heterozygous dominant, right? And then your phenotype. So if you had, like big B is a nose spike and little b is no nose spike, is your dragon going to have a nose spike or no nose spike? No. Yeah, you will have a nose spike, right? So you're going to write your phenotype here. And then this page is for breeding, so we'll have to do that later. But what you get to do is you get to draw your dragon for me right here. Ooh. This is your parent. This is the one that you're building today with those alleles, your parent dragon. Sooner or later, you're going to have a baby dragon, but we're just going to start with the parent. So You're gonna put your alleles together, find out what their characteristics are, and then you're gonna draw your dragon. So you can make him look however you want to make him look, so long as he has all those phenotypical characteristics. So he has a nose spike or no wings, or it has, what does it have? Spots on the neck, horns on the head, fangs or no fangs, breathes fire, does not breathe fire, has 4 toes, has 3 toes, right? So that's what you'll be working on. So tuck this in your binder so that you've got it. And then pull out lab 8 and 9, because you're gonna need both of those today. Pull out 8 and 9, and then I'll call in the rest of the class. Excellent. All right, let's see. And this. All right, gang.